we have the single-cell suspension and master mix on ice.
We also have our gel beads, which have been equilibrated to room temperature for at least 30 minutes. We also have our chip holder and gasket.
First, we will attach the gasket.
Ensure the holder lid is closed. Align the notch with the top left-hand corner. Attach the gasket by holding the tongue and hook the gasket on the left-hand tabs of the holder. Gently pull the gasket toward the right and hook it on the two right-hand tabs. Do not touch the smooth side of the gasket. Do not press down on top of the gasket after attachment.
Next, we will place the chip in the holder.
After removing the chip from the sealed bag, use the chip in less than 24 hours. We will hold the chip by the edges, making sure not to touch the bottom of the chip. We open the Chromium X chip holder with the gasket attached. Please ensure that you are using the Chromium X chip holder. Align the notch on the chip in the upper left-hand corner and the holder. Slide the chip to the left until the guide on the holder is inserted into the chip. Depress the right-hand side of the chip until the spring-loaded clip engages. We are ready to load reagents into the wells.
Let's review the sample loading guidelines.
Up to 16 independent samples can be run on the chip, but only an even number of reactions can be run on the chip. Corresponding wells in rows 2A and 2B of the chip should both be either loaded with independent samples or a mock sample, or with glycerol. For an even number of samples, load the independent samples, master mix plus cells, in pairs in rows 2A and 2B.
In this example, we are loading 16 samples.
We will load 8 samples into row 2A and 8 samples into row 2B. For an odd number of samples, load the unpaired sample in a well in row 2A and a mock sample, which is composed of master mix and water, in the corresponding well in row 2B. While a mock sample can be run for the unpaired sample well, this will result in the loss of this single gel bead reaction.
In this example, we are loading 11 samples.
First, we will load 50% glycerol into the unused wells. Then, we will load 6 samples in row 2A and 5 samples in row 2B. Finally, we will load a mock sample, not glycerol, in the corresponding well in row 2B.
Today, we are running 10 samples. We will load 50% glycerol into the unused wells.
Ensure the lid of the assembled chip holder with the gasket attached is open and is flat for loading. We will add 130 microliters of 50% glycerol to the unused wells in the row labeled 1. When you pipette into the chip, gently touch the pipette tips on the bottom surface and slowly pull up the pipetter as you are dispensing. Next, we will add 140 microliters of 50% glycerol to the unused wells in the rows labeled 2A and 2B. Finally, we will add 140 microliters of 50% glycerol to the unused wells in the rows labeled 3A and 3B. Do not use any substitute for the 50% glycerol solution.
We loaded 50% glycerol into the unused wells. Next, we will combine the master mix, water, and single-cell suspension.